And I'm
This
going to talk to you
conference
about
will now be recorded.
I'm going to talk to you about some of the early work we did on humanized mouse models. Um, so this is going back to about 2016, 2017. We ran it maybe all the way up to 2021. Right. So when we talk about humanized mouse models, when we first came to these models, there were the two options. You could go with the PBMC humanized mouse models. You could go with the the human stem cell humanized mouse models. There was the BLT, so that's bone marrow, fetal liver, and thymus, but that was quite specialized, and there were lots of ethical hurdles to overcome to use those models. So we focused on the PBMC and the stem cell models. And it quickly became evident that with the PBMC models, we had quite a short experimental window because the animals developed GVHD. Although if we went with the stem cell humanized mouse, we had much longer windows, so very little GVHD up to a year. And that's because the human stem cells, the T cells develop, are selected on mostly on murine MHC. There can be some self-selection, but it's mainly murine MHC. And these models were attractive to our bioscience colleagues because they didn't need to generate mouse surrogates for their syngeneic mouse models. They just needed their lead human molecule, and they could go straight into these mouse models. There was also the transgenic option you could go for, but that took much longer to develop those newer models. And as targets changed, you needed new transgenic mouse models, and the field was moving very quickly. So that's where we were. We began looking at the stem cell humanized mouse model, and we thought, well, they take quite a long time to see GVHD. Would our IO therapies break that tolerance? This is some work here looking at GVHD that was done by Alba and my team in collaboration with the University of Cambridge. And you can see here on the left-hand side, this is looking at GVHD. And if the top plots are, we looked at adult bone marrow stem cells and umbilical cord stem cells, and mice humanized with those, and we just after engraftment, they're very stable. We don't see GVHD, and then compare that to the black and the orange plots on the left, which are mice humanized with PBMCs and also splenocytes we had as an option, and you can see the rapid development of GVHD. And that gives you a very limited window in which to conduct your studies. The green plot was something more recent. That was looking at the MHC class 1, class 2 knockout NSG mouse from Jackson Labs, and that gave us a better, longer window to do our studies because it delayed GVHD, but you still saw GVHD. And if you look on the right-hand side, you can see expansion of human human cells in these mice models. With the PBMC and SBMC, there's a rapid rise in human CD45 positive cells driven by GVHD mediated expansion. With the stem cell humanized mice, there's delayed engraftment in comparison. You have to wait much longer. And then that's the pink plot. You can see stable engraftment. With the bone marrow, the adult bone marrow cells, the engraftment's not so good. Adult cells aren't as pluripotent, and so you're much better off using those fetal stem cells. And if you look at the SPMC, the splenocyte humanized double MHC class 1-2 knockout mice, you can see the expansion of those T cells isn't as great as compared to a normal NSG mouse because they haven't got that GVHD driving expansion, although after about 10, 12 weeks, you do see a loss of cells, mainly loss of the B cells. We don't see that in the PBMC standard NSG models because the mice are all dead. So recently there's some work published showing that you could see acceleration of GVHD with anti-PD-1 or anti-CTLA4 antibodies. This is the PBMC humanized mice. So on the left-hand side, the PD-1, you've got the controls in purple, and you can see there's a shift to the left with different doses of an anti-PD-1. And on the right-hand side, you've got CTLA4 treated mice. So in blue, got anti-CTLA4, and you can see there's some acceleration of GVHD. compared to isotype control or a CTLA-4 fusion protein. And when we were looking at which models to look at potential for accelerating this GVHD, obviously with the PBMC models, we thought that would be too fast to sort of tease apart subtle differences between different IO therapies or combinations. Whereas with the stem cell humanized mouse, it took so long to see GVHD, thought that would be too slow. So we thought we'd go for mice humanized with donors that were predisposed to GVHD. There were a couple of publications, one's by Sontag, one's by Greenblatt, and they had looked at different donor HLA types, and they saw ones that had delayed GVHD and ones that had accelerated GVHD. And so based on those publications, we thought we'd use mice humanized with donors which are HLA CW602 or CW401 expressors. So we could reduce the variability and skew our studies to seeing the actual GVHD. And so that's what we did, working with Jackson Lab for the selection of the mice and Crown Bio who run the studies. Now, the molecules we decided to look at initially were an anti-PD-1, an anti-CTLA-4, a bispecific, and a combination of a PD-1 and a CTLA-4. And if I go to the study designs, the first study we had 15 mice per group, 4 donors, and we subsequently increased that to 20 mice, so you can have 4 donors, 5 mice per group. We found that was reduced the variability quite a bit, and we stuck with that. It was just a bit tight with just 15 mice per group. We compared, we had our control, our vehicle control, PD-1 monotherapy, CTLA-4, and a bispecific. And then in the follow-up study, we wanted to compare the bispecific and the combo with the control. And so that was 3 groups. And this, the bottom, you can see our study design. We're dosing every 3 days. We had initially, we're planning on 12 doses, but we had shipped a couple of extra doses to the CRO, and as it turned out, things started happening towards the end of the study. So we went with the 14-dose study, and then we've stuck with that ever since. Now, let's see what they did to the GVHD. So this is what we saw. So the controls in black, see very little. We lost 1 mouse out of all of those groups. We didn't see really much, nothing significant with an anti-CTLA-4 in the stem cell humanized mouse, but we did see something with the PD-1 on its own, about sort of 15-20% response. But compare that to the combo of the PD-1 and the CTLA-4 antibodies together, or the bispecific, which both look very similar. We saw much greater enhancement of GVHD. We also saw expansion of T cells here, and you can see on the right-hand side the combo and the bispecific showed much greater expansion of T cells, as you would see in GVHD, compared to the monotherapies and the controls. We also saw increases in activation markers. We looked at OX40 and CD40 ligand on both the CD4s and the CD8s. You can see with the combo and the bispecific, we see upregulation of those activation markers. Expansion of T cells activate and expression of activation markers. Then if you look in the tissues, we could see GVHD lesions. So on the left-hand side, we got lung, and you could see interstitial lymphocyte infiltration into the lung, portal infiltration in the liver. And most strikingly was the expansion of this white pulp in the spleens of mice, and small macrophage nodules in the red pulp. If you look at the scores, it was interesting. Even in the control animals that didn't show any weight loss, we could see GVHD lesions or pre-GVHD lesions in the tissues of those animals. And similarly, although the PD-1 and the CTLA-4 monotherapies, they weren't markedly leading to weight loss and signs of GVHD, we could see those changes in the tissues before they happen. Then with the combo and the bispecific, obviously we had the highest lesion scores in all mice, irrespective of weight loss, was showing those changes in the tissues. Now, interestingly, we did try to look at the different donors, And all of the different donors we used, one stuck out in particular. There was donor 5443, you can see in red, which was significantly different to all the other donors. And that they developed GVHD much earlier when we pulled the mice that were treated with the combo, the bispecific, together. None of the other donors was significantly different from each other. It was just this one donor. So we went and had a look at their haplotypes. And you can see here circled in the red, donor 5443, and it was the only homozygous donor. All the others were heterozygous. We had thought donor 2315, which had CW401 and 602, may show the most marked GVHD, but no, it was the homozygous donor. So it shows here the importance of the donor MHC in driving that potential for GVHD. Now, we've done a couple of other follow-up studies, and we looked at CD137 agonists. We've also looked at an OX40 agonist. I'm just going to show you the CD137. Very similar data we saw with both molecules. And the study design, again, was very similar. So we looked at the monotherapy, the CD137 on its own, we had the bispecific on its own, and we had the combination. Again, we were treating every 3 days, so a total of 14 doses. And this is what we saw. So CD137 on its own, we saw similar with OX40, very little in the way of GVHD with the monotherapy. With the bispecific, about 50% of the mice developed GVHD during the duration of the study. But then we added in CD137, it just pushed it all the way. And so we had practically all the mice having GVHD. Now, if you break it down by donors, what's quite interesting is that donor 5675 and 2499, the addition of CD137 had very little effect. Just the bispecific on its own could push those donors to GVHD. Whereas if you look at the bottom two plots, donors 5668 and 5674, we were seeing, maybe if we were treated longer, we would have seen the GVHD with the bispecific, but we really weren't seeing much in the way of GVHD in those donors. So it was very donor specific, but you had the CD137 on top, and that was enough to push them over the edge. And you can see the plots in purple, where the CD137 really pushed that GVHD development of those donors. So we got donor effects here. Now, if you look at upregulation of activation markers of CD134 or OX40 receptor and CD137, so you can see that CD137 its own looks to be like a little bit of an increase, but it wasn't significant in CD134, whereas the bispecific really upregulates expression. So then that made sense that when you add these agonists on top, you amplify that effect. Whereas on its own, there's very little expression, so you don't see very much acceleration. And on the right-hand side, well, look at CD137 expression on its own. The drug was on board, so that's why you can't see any CD137 increase. But you can see that the bispecific seriously upregulated CD137. Sorry. And the combo, we can't see that increase because the drug's on board. So that seems to make sense that the monotherapy on its own wasn't accelerating GVHD, but the PD-1 CTLA4 was potentiating it by upregulating that expression. And here, if you look at GVHD scores, you can see that the combo, so that's the PD-1 CTLA4 bispecific with an CD137 agonist, accelerated GVHD even further and pushed up those GVHD lesion scores. One interesting thing is we were interested in liver tox with the CD137 in these mice. We didn't see it on its own, obviously because the CD137 was upregulated without the bispecific. But in the combo, we did see upregulation of liver enzymes. We only saw it with the CD137. we saw the same acceleration with the OX40 agonist, but not the increase in the liver enzyme. So that was a nice observation, given that CD137 agonists have been linked with liver toxicity in the clinic. So that was the sum of the humanized mouse studies we've been doing. So you could see that as we move to these models, and we look at combination therapies or complex bispecifics, those studies are going to be limited by acceleration of GVHD, which is going to limit our experimental window and the interpretation of histology. Is it GVHD driven, or is it a drug effect? We know donor HLA type can modify GVHD development in these humanized mice, and our bispecific molecule really didn't show any marked potency increase over combination therapy. While these GVHD endpoints can be useful, possibly to evaluate potentials of different combinations, what we have done more recently is we've moved away from this model and focused more on the MHC class 1, class 2 knockout mice, which can delay that GVHD and give us a better experimental window. And we're using sort of PBMC CDX models based on this class 1, class 2 knockout mouse model now to evaluate our therapeutics. But all that previous work was very informative in guiding us to that path, and understanding the limitations of these models, these humanized mouse models, particularly development of GVHD. And with that, I'd just like to thank Alba, who's was my postdoc at Cambridge, who's working on humanized mouse models, and is now in our team working on those models for CAR T cells. Jean-Martin was the pathologist who did all those sections, and Sean Maguire was my study monitor of running those platform studies. Then our bioscience colleagues, Matt and Simon, and our antibody engineering colleague, Gareth, have all contributed to this. Studies were run at Crown Bioscience, and we collaborated with Jackson Labs on these mice as well. So with that, I'll stop and I can take questions.